So the glucose went through glycolysis, and the glucose produced two 3-unit carbon units called pyruvates. Pyruvates went through the PDC, the pyruvate dehydrogenase complex, to produce acetyl-CoA. And the acetyl unit was brought in by the coenzyme A to the Krebs cycle, where the two carbons were lost and NADH was made and FADH2. Now, what is the use of the production of NADH and FADH2? These are molecules, electron carriers, carrying electrons. They are reduced in this form, and they've been being made from the glycolysis, the PDC, and every step of the process. We are making NADHs and FADH2 only in the Krebs cycle. What is the purpose? I likened them in my earlier videos to checks. They carry value, but you cannot really go to a store and use them to buy anything. ATP, when it's produced with a substrate level phosphorylation, can be used. It is the cash of the cell. However, NADH and FADH2 cannot be used. What is their purpose? Now, this video will explain to you what the purpose is. This is a video about the electron transport chain and chemiosmosis. Now, here we are in the mitochondria. We are located in the mitochondrial matrix. I was hoping that the Krebs cycle here will hint where we are located. So this is the matrix. And just to review, this is the inner mitochondrial membrane. This is the outer mitochondrial membrane. This is the intermembrane space. And on the inner mitochondrial membrane, we have a series of proteins, which shall remain nameless for the purposes of grade 12 in this video. And so what happens is the NADHs that have been produced in the PDC, the pyruvate decarboxylation reaction, and in the Krebs cycle, come to the first complex. And at the first complex, they drop off their electrons. So I'm going to write down NADH becomes NAD plus by losing the two electrons. The two electrons go into this complex, and they cause the complex to change and to shift its form, and thereby pumping out protons. We're gonna put four protons into the inner membrane space. Now the electrons are picked up by the second complex and brought in here. And when the electrons get to this complex also changes conformation and pumps out 3 protons. And this carries on. This protein comes in and picks up the electrons from this complex, bring it to the next. And now the electrons are being transferred. So the electrons were transferred from this one to this one, from this one to this one, and here the electrons are here. And this complex also changes conformation and causes the pumping out of 3 protons. I don't know what you're thinking about this. What is the purpose of pumping all these protons into the intermembrane space? Well, this will become evident in a few minutes. Hopefully less. Now, this protein is saturated with electrons, and it cannot receive any more electrons unless somebody comes and removes these electrons. So oxygen is our final electron acceptor. Oxygen takes these 2 electrons plus 2 protons and produces 2 molecules of water. Now, I'm going to show you this later in the overall reaction for cellular respiration. So the oxidation of NADH to NAD plus caused the pumping out of 10 protons. So I'm going to write that down here. NADH caused the pumping out of 10 protons. And we're going to see why they're so important later. 10 protons. Now, what do we do with FADH2? FADH2 does not go to the first complex. Instead, it goes to the second complex, and it is reduced, oxidized at the second complex. So the second complex brings this electron to this protein to cause the pumping out of 3 protons, and this protein to cause the pumping out of another 3 protons. And remember, it's not just the 10 and the 16 protons. We have so many FADH2s and NADHs being produced in this process. Now, all of these come and get oxidized, and we need more oxygen to come and be the final electron receptor. And here, after this entire process of the oxidation of these electron carriers, we have built up, or not we, but the mitochondria have built up a huge concentration, a very high proton concentration in the inner membrane space. And because of that, we have very low concentration of protons in the mitochondrial matrix. So here there's a very high concentration, and here there's a very low concentration. The protons are dying to just come back in here because they really want to go down their concentration gradient. To no avail. There's no way they can come back here. There is no way. To come back here, they cannot. The only way to come back down their concentration gradient into the mitochondrial matrix is through this last molecule or complex called ATP synthase. Now, we built up this huge proton gradient, very high proton concentration here, very low proton concentration here. The protons are dying to come back in, and they are forced to go through ATP synthase. As 3 protons go through ATP synthase, okay, this causes the ATP synthase to rotate. We're not going to talk about the mechanism now, but as it rotates, it will take an ADP, adenosine diphosphate, plus an inorganic phosphate, and it's going to produce ATP. So let me reiterate this. In every step of the process of cellular respiration, NADHs and FADH2s have been made. Well, I'm going to say that FADH2 is only for the Krebs cycle, but electron carriers have been reduced. Why? Because they're going to come to the electron transfer chain, they're going to be oxidized, and this oxidation is going to produce this proton gradient that is going to lead to the phosphorylation of ADP to form ATP. This method of production of ATP is called oxidative phosphorylation. Oxidative phosphorylation. So oxidative because it is due to the oxidation of the electron carriers, and phosphorylation because the ADP is being phosphorylated. This ATP synthase is adding a phosphate group onto the ADP to make an ATP molecule. Oxidative phosphorylation, as you see, is different than a substrate level phosphorylation. Oxidative phosphorylation is an indirect process. It's the production of electron carriers that will later on be oxidized to create the proton gradient that's going to be used to produce ATP. Now, as you can see here, the oxidation of glucose is linked to the phosphorylation of ADP. Okay, this process is coupled. The proton gradient that we're producing here is coupled to the ATP production. All right? So if there is anything that interfered with this inner membrane and allowed the protons to go down their concentration gradient by other means other than the ATP synthase, then this would cause the production or lead to the production of less ATP, because we're dissipating, it's the molecule, it would be dissipating the proton gradient. It is the proton gradient that's forcing these protons to go down into the ATP synthase or through the ATP synthase to make this ATP. If there's anything that uncoupled this process, okay, let's say there's a molecule here that allowed the H pluses to go in, that would be a problem, because now the proton gradient is no longer coupled to the ATP production. This type of molecule is called an uncoupler, and it used to be a main ingredient in one of the diet drugs administered well, 100 years ago, around that time. Obviously, this drug has been discontinued because a lot of people died from consuming this drug because their brain was not able to produce as much ATP, and their brain cells died. Anyways, in our next video, I will be talking about the ATP count from substrate level phosphorylation and from the oxidative phosphorylation. I hope you enjoyed this video, and if you did, please subscribe to my channel. Bye!